One of my pet peeves is to call bacteria commensal because they're not just there. They are doing something. What are the bacteria doing and how can we manipulate them? There's more out there definitely that we don't know than we do. Just opens up this whole area of research that can keep somebody busy for a lifetime.
Welcome to Unplucked, the official podcast of the Poultry Science Association. This is the podcast where we strip away the fluff and get real about poultry science from bench to barn. I'm your host, Andy Vance, Executive Director of the Poultry Science Association. This week, after decades of pioneering gut health research in poultry, we're joined by one of my favorite members of our Poultry Science Association Board of Directors members, Dr. Lisa Bielke. From Arkansas to Ohio and now North Carolina State, Professor Bielke talks about a deep focus on turkeys, emerging challenges in controlling Eimeria and Histomoniasis, and why gut health is more than just a buzzword. Whether you're managing flocks or advancing science, this is an episode you don't want to miss. Dr. Lisa Bielke, Prestige Distinguished Scholar of Turkey Health and an Associate Professor at NC State University, did her undergraduate studies at Texas A&M, earned a master's and a doctorate in poultry science from the University of Arkansas. Her research at NC State focuses on improving gut health in poultry and immunity through feed additives, vaccination, probiotics. Her labs developed an internationally licensed oral vaccine targeting key poultry pathogens. And she's also been recognized in a variety of different venues with a number of honors, including the Evonik Award for Achievement in Poultry Science from PSA, the PSA Early Achievement Award, the Arkansas Biosciences Institute Early Investigator Award early on in her career, as well as the Highline International Research Award and the Novus Outstanding Scholar Award. She's done great work everywhere she's been along the way, and as I mentioned, serves as an active member of our board of directors, in addition to being a section editor for our flagship journal, Poultry Science. All right, Dr. Bielke, as I mentioned, you've been at a few of the most prestigious institutions in our community through the course of your career. Of course, you and I first got to work together when you were at my alma mater, the Ohio State University. So I was a big fan of your work then and now. How's your research focus evolved as you've moved from your undergraduate days in Texas to doing graduate work at Arkansas to faculty staff positions at Ohio State and now NC State? What's that look like for you as your research program has evolved and developed throughout your career?
So it's mostly come down to what the industry needs and kind of keeping an eye on that. In undergraduate and even my graduate career, one thing I was taught was to try to stay ahead of the problems and become an expert before the industry needs an expert. So, it's one reason people see me all over the place going to conferences. I'm trying to hear what people are dealing with and stay ahead of that and talk to allied industry and go to some of the more regional and local events where I can learn what industry is doing. So I would say my research career has evolved as the needs of the poultry industry have evolved.
I mean, it's pretty great practical advice for a career more generally, but especially in a field that is constantly evolving. Poultry production or systems are constantly evolving to meet the needs of consumers, what they're concerned about. Obviously, bird health changes and evolves pretty rapidly. So I want to drill in on this idea of gut health, since that's been one area where you have built quite a reputation. What drew you to that specifically in the first place? Was it a problem that you were hearing people talk about more, or was it something that as you were under the microscope and said, this is kind of fascinating, and just piqued your natural curiosity? What got you into that space?
It goes all the way back to probably my master's degree. One of the earlier labs that were working on this idea of selecting probiotics for specific function. And we already knew probiotics were good, but we selected probiotics. One of the first labs probably to do it in poultry, not necessarily globally, but in poultry, where we could select a probiotic to have a specific function. And in this case, it was against salmonella, to reduce salmonella in birds. But one thing we noticed doing that was that even in selecting something to help reduce salmonella in actually turkeys, it actually gave them a lot of other health benefits. And so that really started this whole okay, bacteria are actually in the gut for a purpose. They do a thing. One of my pet peeves is to call bacteria commensal because they're not just there. They are doing something. And so I really piqued my curiosity into what are the bacteria doing and how can we manipulate them, not just with probiotics, but in other ways. And then that just opens up this whole area of research that can keep somebody busy for a lifetime.
Something you said earlier about becoming an expert in the problems before they're a thing. The timing of your career is really interesting in terms of how the industry has looked at antibiotic usage. So we kind of went from this area where kill them all and let God sort them out with regard to these bugs. And now we have maybe a more nuanced understanding of bacteria, the good ones, the bad ones, are they just hanging out there? My question for you though is how much do we still not understand with regard to what those bacteria are doing? As you said, they're there for a reason. They're not just hanging out at the bar, but what do we know versus what don't we know yet? How do you feel we are developing our overall understanding of the role that these bugs play in the gut?
I would say we definitely do not know more than we know, right? So that's the way it is in a lot of science. But one of the big things for us is that the big data, the omics technologies have really opened up what we can do in terms of gut health, just wide open. Because now we're not just trying to culture bacteria and run these little tests or do multitops or whatever and try to figure out what bacteria are there. We can just run genomic sequencing and get this whole list of things, some of which we don't even know what they are. They're uncharacterizable. Nobody's been able to characterize them before. And then beyond just knowing what's there, we can use omics technologies to know what they're doing. We can taxonomically profile those genes for function and figure out what those bacteria are doing, but you can pair that up with what's happening in the bird and see these changes in that host relationship in a way that we've never been able to do before. And so in the last decade, our abilities have just blossomed. It's just been, as Walter Bocci told me the first time I ever did an omics study, he said, be careful, it's going to be a data tsunami. I didn't understand what he said at the moment, but now I do. There's just so much there that we get more data than any one person or one laboratory really can sort through. And then on top of that, most of the times when we publish it, we're required to deposit it into a public database. So that puts our data out there for anybody that just wants to go through the databases to even acquire more data. There's more out there definitely that we don't know than we do. We're just barely scratching the surface. And probably every time we do an omics study, we ask more questions. We find more things that we want to pursue beyond what our original goal was.
On the one hand, I'm thinking as it would be a researcher if I was pretending to be in the Bielke lab for a minute. On the one hand, like great job security, I'm always going to have something to study next. In theory, there's always more papers to write. There are always more data to sort through. How do you prioritize or how do you evaluate, all right, what is the next question? Is it very logical, methodical, or do you see a squirrel in there every now and then be like, oh, wow, I didn't expect that. Let's go chase that one down. Like, how's that go?
Yeah, there's definitely some squirrels. I do a lot of wrangling graduate students back onto the task at hand to finish what we started. And then there's always the, like, if you want to pursue this later, we can. But in terms of deciding what we're actually going to do whenever we want to do a study, especially these big omics studies, usually it boils down to poultry health, right? It evolves, but usually we'll go into it with a very specific health related question. Like right now, we're looking at this vertical transmission of bacteria and how the breeders are affecting their offspring. And I've seen some unexpected stuff there. So it's like we finished that study and we answer that question. And now 3 or 4 years later, I've moved on and used actually that study that we completed as preliminary data for the next thing that we're going to do for probably the next 10 years. And we're going back to that old data and looking at it and sort of mining it to see like, okay, this is our starting point. What can we use out of this to keep looking for more funding or to establish the basis for our next studies? So it definitely keeps you busy. Like I would say every time we get some data, you can go back to it and look for something else to do. If you don't have any ideas, just go make some graphs and run some statistics and you'll find something. Just stare at your PCOA plots for a minute.
Yeah, that's one of the things I always find fascinating when I'm talking with researchers is just how they approach the next question and what do we do with what we've learned. So you talked a little bit about your work with turkeys. A lot of times, I think when we're talking about poultry industries, we just sort of default to broilers, or if we've been talking about high path a lot, we sort of default to layers or maybe the center of gravity. When you work with turkeys, what are some of the unique challenges in the gut that make that research different, more challenging than what we would see in the broiler space as a default setting?
In some ways, turkeys are just kind of a big chicken, and there's a lot of overlap between the two. So I can take my experiences in broilers and knowledge from broilers and apply that to turkeys, generally speaking.
Hmm.
But they do have a lot of things that actually are not the same. Their behaviors are different. So if we're going to talk about controlling a disease, you can't just say, okay, we're just going to treat it the way we do broilers, right? Like even Eimeria, we're finding vaccinating turkeys is not quite the same as vaccinating chickens. And so we're seeing some difference. It's some to do with the Eimeria specifically that infects them, but also we're seeing some difference sort of in their immune responses and how it cycles. And so that definitely, you can't just take the broiler stuff or the broiler breeder stuff even and apply it to the turkeys. Some of it's also just the nature of the production system and how it applies. Because turkeys these days, they go into the 6-week brooder houses and then they get moved to a completely different farm. But they're also a lot longer lived than a broiler is. So we're trying to get hard and fast vaccination with broilers, whereas turkeys, you may not need to go so hard and fast because you're at a higher risk of causing mortalities and overt disease from your vaccination. It's harder to manage that way. So you can maybe step back and manage it a little bit differently and play a longer game.
Well, you mentioned the difference in the production systems and management of the birds for the obvious reason they're much different sized animals. And like you say, behaviors might be different. I'm also wondering about geography. How much of a role does where the production happens play into this question of gut health? Are the bacteria or other things in that GI tract different in Arkansas than they are in North Carolina, than they were in Worcester, Ohio, than they are in the upper Midwest where the turkeys are? Like, what do we know about that and how different the critters that we're dealing with are depending on where in the world they are?
Yeah, parasites are definitely different regionally. In Ohio, when I was there, it wasn't as big of a problem regionally to worry about Histomonas. People kept asking me if I would do Histomonas work in Ohio. And it's like, well, I'm not really set up to deal with it. That's not a problem here. I'm not getting a lot of pressure to do it. But within a week of stepping foot into Scott Hall at NC State University, people are saying, what do you know about Histomonas? So definitely some regional diseases there. The North definitely has the advantage of insects die off
Sure.
in the winter. And so even some of their bacteriological diseases, well, and the parasites that are carried by insects becomes a little bit more seasonal for them and not an all the time thing to deal with. So you probably see that in all production systems, but turkeys tend to be spread out a little bit more, whereas broilers are a bit more concentrated. So they do have some regional problems.
Okay, so we've talked about Eimeria and Histomoniasis. You guys in your lab, in your research focus, work a lot with those two. You also look at pathogens that are more, what do I want to say, centrally involved in discussions of food safety. You've got some great grad students working on a lot of different things in that broader space. What are the projects you're working on right now that get you up in the morning and have you excited to come to the office and come to the lab every day?
It's going to be more the vertical transmission of bacteria. That's the one where I just kind of find myself wandering through literature when I've not wanting to focus on the things I'm supposed to be doing. I'll just kind of wander through and look for papers and new work on this vertical transmission, mostly because what I think I'm seeing is that what comes from the parent stock not only affects sort of their pioneer colonizers that make this established microbiome within the gut that can be good or it can be bad, but it also affects how much we can change it. So we always hear about probiotics failing and they don't do anything. And why are they not working? Because they used to work over there, but now they're not working over here. What's different? That might actually be one of the things that are different. And we've looked at that in more than one species of poultry, and it looks to sort of be universal. And so, like, how can we affect that? What do we need to be there? And how can we ensure that it's there? That's one. If people want to know what I'm excited about today, that's the one. Salmonella always gets me excited because it's not just a food safety pathogen. It also affects inflammation in the gut, which leads to the whole gut health thing. You can say, well, we only need to care about salmonella when the birds are going to the processing plant because it's a food safety pathogen. But actually, there's a whole lot out there that says that it affects their health. It's not just a human health issue. And it's subclinical, but it can make the birds more susceptible to other diseases. It can affect their feed conversion ratio. It can affect bone density. It goes way beyond just being a food safety pathogen. So anytime someone wants to say, hey, what can we do with Salmonella these days? I'll have a list for them.
I love it. So that raises a good question. That this is a notoriously complex area of bird health. Is this idea of gut health, right? And so measuring it is notoriously complex. You just gave some great examples of, it's a probiotic issue or salmonella and what it actually does in the gut. So what are some of the tools, indicators, technologies that you're using or maybe that you're developing to help manage or make that process more accurate, measurable, workable? What's that like today?
Well, so inflammation is a very complex disease. And so what are we in now? 2025. So maybe 15 years ago, I set out being a little bit naive, thinking we're going to set up a few ways to measure inflammation and we're going to have all the answers we need. And it turns out it's not that simple. So we've been trying to find other ways to characterize inflammation. Fortunately, it tends to be a fairly universal problem. So we can use human medicine or medicine from other animals and try to adapt it and apply it to poultry. It's not always super straightforward. A decade ago, we didn't have some of the genes characterized that are needed, but it really boils down to trying to find ways to measure inflammation at sort of the applied level and the way that it's affecting the animals instead of looking for specific genes and having all of these interleukins and TNF alphas and all of these numbered compounds that have effects, but they're part of a cascade. And so I'm always looking for, okay, what's the end effect on the animal and how can we measure that on the animal? Because once we know how it's affecting that animal specifically, then we can start to look for a solution. If we could do it with a blood draw, that makes it easy for producers to use it instead of us having to actually collect tissues because those become terminal samples. A lot of times we'll start out with collecting tissues and then try to figure out how to apply it more towards a blood pathogen way. So I've always got something I'm working on. I'm actually working on a grant right now on a new thing that will help us perhaps measure how we're sort of aging birds as they go later in life. And that would come alongside with perhaps our breeding stock and our layers and how to potentially help them stay in production longer.
Longevity is another idea that's fairly complex. So I'm interesting to hear how that comes along. That's exciting.
It's still just a little bitty embryo in my brain, so we'll see how it comes out over the next few years.
That's how they all start, right? You had an idea or somebody brought up an issue or presentation you heard at a conference, right? Back to something you said earlier on. So when you look at research, and a lot of the examples you've used already, Salmonella is a great one. Longevity could be one in this space. But you're looking at research that spans, say, performance issues, how we manage the bird, to some of these that have food safety goals attached to it. Salmonella, as I say, is the easy example. When you're looking at research like that might span a few different areas of the production and value chain, are the goals there, do you find them typically in alignment, or do you have to sometimes sort out what's the most important issue here? I got to deal with the food safety issue and I'll worry about the production problem later. Or, hey, I need to get the production issue solved and we'll figure out the food safety implications later. How do you sort through that as a researcher, getting the different goals in alignment for the different chains of the production system and who needs what from us as scientists?
I ask myself, what's the end product out of this research? Sometimes we don't know what the end product is, but I've had a lot of really good ideas. And then I sit here and I think, or I talk to some colleagues and I pass the idea around a little bit and I start asking, well, what are we going to do with this information? Right? Sometimes we don't know what we will do with the information, but if I can't even say what the next step with that information would be, then I'm probably not going to pursue it. But I definitely always try to say, okay, what is the poultry industry going to do with this? If I go to a turkey producer, how are they going to use this information to make a better bird in whatever way they want to use it, right? But even if I can't get that far, I'm always asking myself, what will we do with this information at the end of this research project? And if there's not an answer, we're probably not going to do it. If there is an answer, then it's probably worth pursuing.
That's a great approach. Begin with the end in mind. I mean, that's always one of my favorite Stephen Covey Seven Habits things. I think that's
Right.
that's such a smart way to tackle that. When we start then moving, that's a good segue into something I want to think about is the actual practical implications or industry impact of the work we're doing in the lab. How well do you think gut health research today generally is translating to on-farm practices? We'll say generally, but then maybe most especially in turkey production, since that's a specific area of expertise for you. How well are we doing there at taking that research from bench to barn, so to speak?
I've actually seen quite a bit of what we do get translated and see producers paying attention to it at least and trying to figure out how to make things better for the birds. I do say, yes, begin with the end in mind, but we don't always know the paths to get there. And what I'm doing in the lab doesn't need to be practical just yet. That if it's worth doing, then somebody else will figure out how to make it practical, right? Or try to at least. And so, and I've seen it happen. We've got companies working on making microbeads that can go into feed so that we can help stabilize some of these probiotics or even other acid type compounds and phytogenics and things like that. They used to be very inconsistent because they weren't stable in feed and they would need to go in the water, or they oxidize or something and they're not really stable. Companies are figuring out how to make that happen. So in the one lab, we're saying, okay, this actually really has a benefit. And now it needs to go to a processing engineer or a food scientist or somebody that knows how to implement it. So definitely, I've seen a lot of those things really come around and develop and kind of change my mind even on how I thought about them. I used to think some of this stuff was never, ever going to be used in the poultry industry because it wasn't practical. Well, somebody cared enough to make it practical. So we are seeing things come around, and even probiotics, we've seen it go from just give birds something or anything and you'll see an effect to, well, now we're going to try to find probiotics that have specific functions and try to use a probiotic that will help us solve our needs. You can't just put something out there that does everything. We know that one probiotic won't do all the things. So it's definitely evolved over the last few decades.
All right. So now with that in mind, I want you to pull out your crystal ball. I always like to ask what I call the crystal ball question in every interview. So as you look 5, 10, 15 years down the line, what role do you foresee microbiome science generally playing in the future of poultry health management? Is this going to continue to be a really central, integral part? Is it a lot of hype and fairy dust? Are we just scratching the surface? What's your view of the future for this area of research and how it will influence our management of the birds?
I think we're really shifting from a mindset manipulating the microbiome to control disease to really thinking about manipulating it to benefit the animal, and not just from a health standpoint, but also a nutrition standpoint. We actually rely on bacteria in the gut to do a lot of processing of the food and digesting of the food so that the animals can absorb it. There's enzymes that are produced by the animal, but we also depend on the bacteria. So how can we manipulate them to benefit our birds even more? And what can we do in terms of putting more functional aspects to the microbiota that the birds have? It's not just diseases anymore. It's diseases, health, and nutrition.
Great answer. I love that. And I think one of the things that highlights for me, and you've actually touched on it a couple times during this conversation, the people in different disciplines that you talk to or work with or interface with regularly. I know this summer you've been to, of course, the PSA annual meeting and listened to a ton of talks and presentations there, both in and out of your specific area of direct expertise, so to speak. But you're also were at the American Association of Avian Pathologists meeting and listening to talks there. Give your pitch to maybe it's young faculty members or graduate students who are listening to this or folks in other disciplines for doing that thing that you just said of talking and listening and working with people all throughout this broader ecosystem to solve some of these big, hairy challenges that we have in the industry.
So one of the biggest things that I advise definitely my students and new faculty, if they're asking for my opinion, is go to the meetings, but don't just go to the meeting. Go present something at the meeting. Even if you don't think you have a big oral talk to give, put together something small that can go on a poster and go to the meeting and have something to present, because that's going to get people to talk to you. And you stand up on stage and you give an oral, people will talk to you. They'll see you, they'll have ideas, they want to say things. And you go to the poster, go to the poster session. Don't blow the poster session off. People will come by and talk to you. Even if it's only one or two, you'll get to talk to some people. You'll get their opinions. You'll learn something from them. Go to the social events and talk to people at the social events. Like, don't just go to the meeting to go to a few sessions and listen and then turn around and go do your own thing. Like, if you're present, people will talk to you. So just be in the space with your eyes up. Don't be on your phone. Or, I know we're not all very social people. I'm actually not naturally a social person. I can go do it for a little while and then I need to go back to my hotel room and be quiet for a bit. But it takes practice to get out there. But definitely, if you're going to go to a conference, be present at that conference. Be as approachable as you can be, but also approach other people. I try to be approachable and smile, even if I'm not in a good mood, still say hi to people, because I want to talk to people about what I'm doing. And I know I've probably reached that stage in my life where people have been reading my research and they see me as somebody that has done important things. And they might think that I'm not approachable just because they've kind of built me up in their minds. But I'm actually not that big of a person. Like, I like to talk to people. I want to share my ideas, because if it will help them develop their own ideas, that's great. That's why we're there. I don't know of anybody that goes to a conference just to stand up on stage and give their talk and have everybody just follow their ideas and then walk out the door, right? Like, we want to share it and we want to foster that collaboration and have the talks, because it not only helps us develop what we're doing, but it helps other people further their research. And we're all here for the same purpose, right? We're trying to make raising poultry better and advance the field. So if you go to a conference and you spend $3,000, but it gets you a research contract worth $50,000 or $60,000 with somebody, that's a pretty good return on your investment. Like, find the money to go to the conferences when you're young. Go be there, talk to people, make yourself known. Just put yourself out there. Go do it.
Great advice. And I think, the people that I've met in our community who are the most successful are the ones who do that well and have done so consistently throughout their career, build those relationships, open doors. Great advice. And that's a good place to end it. Dr. Lisa Bielke, first vice president of the Poultry Science Association, a distinguished professor at North Carolina State University. And I will also use that as an opportunity to encourage you to join us in Macau, the Special Administrative Regions of China, this October, as PSA hosts its inaugural PSA Pacific Rim Scientific Conference. Professor Bielke will be on hand there, along with other members of our PSA board, our editorial boards of Poultry Science, the Journal of Applied Poultry Research, and European Poultry Science. Many of them will be on hand presenting lead-off talks and engaging with several hundred members of the scientific community in the Pacific Rim. I look forward to seeing you there in mid-October. You can find details at poultryscience.org. We've talked a lot about gut health this episode, and that's a good segue into reminding you that you can register for our September PSA webinar coming up on September 16th, just under 2 weeks from today. We'll be talking about the impacts of mycotoxins on feed, some research and gaps. Dr. Todd Applegate is chair of the Department of Poultry Science at the University of Georgia and a fellow of the Poultry Science Association. Will be presenting about mycotoxins in animal feed and the challenges those present to poultry health, performance, and profitability. That webinar at noon Central on September 16th. You can register at poultryscience.org. One favor I would ask of you, friends, subscribe, rate this podcast wherever you listen to your podcasts. And remember, if you turn on the notifications, you won't miss an episode. Ring the bell, as one of my favorite YouTubers put it, so you get notified as soon as we drop the next weekly episode of Unplucked. For the Poultry Science Association, I'm Andy Vance.